Thanks, Chrissy, for that introduction. A lot of that you've stolen my thunder, but thank you, and for setting the scene, especially after a talk on commodities, always a bit hard to follow. So thank you so much for being here today and to hear the story of Argenica Therapeutics. We are a pretty newly listed company. We listed on the ASX in June this year and have had a pretty good start, so listing it at 20 cents and hovering around the 36 cent price at the moment, with a huge amount of value still to be had in the company. So just a standard disclaimer before I get into it. So I just wanted to give you a bit of a snapshot of the company. I'm sure a lot of you haven't heard of Argenica Therapeutics. So really what we are trying to do is develop these best-in-class novel neuroprotective therapeutics. So therapies that can protect brain cells following a stroke and other types of brain injury. And we're doing this by developing and commercialising our lead candidate, which is ARG-007. So this is a neuroprotective peptide, it's cationic, it's arginine rich, and it comes with a huge amount of preclinical research behind it out of the Perrin Institute and the University of Western Australia up in Perth. So we've got some fantastic data that shows the efficacy of this drug in multiple models of stroke, but also other brain injuries, including traumatic brain injury and perinatal hypoxia. So we still partner really closely with the Perrin Institute and the University of Western Australia to continue to build that body of research and knowledge around the efficacy and the safety of ARG-007, but also looking at some other drug candidates. And we've recently announced our partnership with Linear Clinical, which is based up in Perth, and they are a clinical trial facility. So we'll work with them closely to do our phase 1 clinical trial. The company has really strong IP, so we have patents granted in the EU, Japan and China, and US national filing is in progress. And we own the IP 100%, so it has come out of UWA and into the company, and we are free of all royalties and encumbrances around that IP. So just our capital structure, we have 37 million shares on issue. A number of those shares are still in escrow from many of our seed investors and also board and management. We just announced this morning our cash balance to end of September is sitting around 5.9 mil, and that will take us all the way to the end of our phase 1 clinical trial. Market cap's hovering around 28 mil as well. So just on to the drug itself and what it does and why it's so important in a stroke model and other types of brain injury. So cell death in the brain, or it's also known as infarction, results from an inadequate blood supply to the affected area. So if we're talking about stroke and if we're talking about ischemic stroke, which makes up around 85% of all strokes, that inadequate blood supply is caused by a clot in the blood vessels in the brain. And so you get this immediate infarct, this immediate area of cell death. And then what happens as time is delayed to treatment, that cell death essentially cascades out. So you get this ripple effect of cell death happening in the brain. So whilst there's no drug that can actually stop that initial infarction or that initial area of cell death, what our drug has shown to do in preclinical animal models is to have a multiple mechanism of action that stops that cascading out of cell death. So in animal models, we've shown that we can slow that progression of cell death and preserve that still viable tissue. And that is really important in functional recovery for these patients. So we are going after the stroke market initially. That is where the majority of our preclinical data is. But we also have some fantastic preclinical data in traumatic brain injury and also perinatal hypoxia. So I'll talk about those two a bit later. So why stroke? Why are we going after this particular indication? It is one of the leading causes of death and disability worldwide. So 15 million people have a first stroke globally every year. And as Chrissy said, that equates to one person every 19 minutes in Australia having a stroke. Only about 10% of those will make a full recovery. The majority of people that suffer a stroke have a long-term disability, lasting brain damage, and of those 15 million people, around 5 million people will die because of the delay to get to treatment. So we know that time is brain. So for every minute delayed in receiving treatment, 1.9 million brain cells will die. And that just expands out. So for every hour stroke treatment is delayed, your brain ages 3.6 years. And there is currently no universal drug on the market that has the ability to protect brain cells following stroke. So we hope to address this market with ARG-007. So for those of you that aren't familiar with, I guess, what sort of the current treatment plan is for someone that's suffered a stroke, when you have a stroke, the ambulance arrives and there's very little that paramedics can actually do to triage that patient. They'll determine the severity of the stroke, doing what's known as a RACE score. But the aim is really to get that patient to hospital as soon as possible, diagnose a stroke and get them into treatment. It's so critical to diagnose a stroke because there's two main types of stroke. So ischemic stroke, which is caused by clot, and then there's a hemorrhagic stroke, which is caused by a bleed on the brain. So for an ischemic stroke, the treatment is really to remove that clot. So you can do that either by administering a drug, which dissolves the clot, or by mechanical thrombectomy, actually surgically removing the clot. But if you give that clot-busting drug to someone that's had a hemorrhagic stroke, it'll exacerbate the bleeding of that stroke patient and can result in death. So it's really, really critical that that patient gets to a stroke care unit and actually gets diagnosed a type of stroke. So in all that delay in that treatment, you're getting those rapidly increasing levels of neuronal cell death, which is proportional to the functional outcome of those patients. So what we want to do with ARG-007 is have it administered by paramedics in the field to stroke patients so that we can start protecting brain cells shortly after someone has suffered a stroke. This buys them time, essentially, to get to hospital and receive the treatment that they need. So we're reducing that total amount of cell death, preserving still viable brain tissue, allowing that patient to have a better functional recovery. So the real benefits of ARG-007 is that it's simple. It's a simple IV injection. So it's a one-off injection infused in the patient over 10 minutes. We know from all our extensive animal studies that it does reduce brain injury and neuronal cell death. It fits very easily into the current stroke treatment plan, so it can be administered by paramedics, emergency physicians. And it will extend that time window, buying these patients more time to get to hospital and to get the treatment they need. So if you think about patients coming regionally, having to get Royal Flying Doctors, coming into either Charlie's or Fiona Stanley, they need that time. They need to preserve those brain tissue. And ultimately, it will improve the lives and lower costs to the healthcare system. So we've got really encouraging results to date for ARG-007. So as I said, extensive amounts over a decade of preclinical work undertaken at the Perrin Institute. We have shown that we get a 67% reduction in brain tissue death following a stroke, and we see that protection lasting up to 28 days post-stroke as well. We also know that our drug does not exacerbate bleeding in a hemorrhagic stroke patient, so we can administer it in the field by paramedics. It will not impact hemorrhagic stroke patients. And we've also seen no adverse effects in animal studies of our drug. And one of the critical things is that we know that our drug, when it's co-administered with the clot-dissolving drug, is not degraded. It remains stable and continues to provide neuroprotection. So patients can still receive the clot-busting drug through their treatment plan. So just some of our near-term catalysts and milestones that we're looking to hit sort of towards the end of this calendar year and into next calendar year. So it's all really gearing up for our phase 1 clinical trial. So for those of you that don't sort of understand the clinical development pathway, the phase 1 clinical trial is looking at the safety of our drug and the tolerability of our drug in healthy volunteers. So it won't be in stroke patients, it will be in healthy people. So in order to do that, there's a number of studies that we have to do to get there. So we have to look at the pharmacokinetics of the drug. So how is it being absorbed, distributed, metabolised and excreted in the body? What's actually happening to that drug in the body? We also need to look at the safety and the toxicity of the drug as well before we can move it into humans. So we've initiated those 2 types of studies. We've also initiated the work with Linear to set up for our phase 1 clinical trial, and we hope to start our phase 1 clinical trial towards the end of Q1 next calendar year. So this is our proposed trial design. So I will caveat that this is still subject to ethics approval, but we will have 32 healthy patients in the phase 1. We'll split them into 4 cohorts, with each cohort receiving a different dose of the drug. So we'll start the first cohort, which will have 8 participants receiving the lowest dose of our drug. These cohorts will be done sequentially. So we'll bring the first 8 volunteers in, 2 will receive a placebo and 6 will receive ARG-007. We'll make sure in that lowest dose that there's no adverse reactions and then move on to the next dose. So volunteers come in, they spend 48 hours in Linear. We monitor them, we take bloods, we assess vital signs, those sorts of things. Take further data at day 3 and at day 8. So we will have preliminary data coming through as we progress throughout our phase 1 trial, which we will put out to the market. Thanks. So really the primary objectives of the phase 1 is to understand how ARG-007 affects the body, what's it actually doing to the body through those PK assessments. Make sure that it's safe when it's administered through the IV injection, and then determine what that ideal safe dose is across those 4 cohorts, and obviously ensuring that there's no adverse reactions in any of those volunteers. So once we complete our phase 1, we'll be progressing to our phase 2. So this is where we do it in patients that have suffered a stroke. We'll likely do that in the emergency department, and that will be in collaboration with stroke care units across Australia. So in Perth, you have a stroke care unit at Charlie's and at Fiona Stanley, so we'll bring them on board, and then other stroke care units across Australia. But what is great about doing a phase 1 and looking at the safety of your drug is that you can accelerate the progression of that drug in other indications. So as I mentioned before, we'll look at perinatal hypoxia, which is essentially a reduction in oxygenation or blood flow in infants. So it usually happens immediately prior to, during or post-labour, and it is one of the leading causes of infant mortality and disability worldwide. We'll also progress our traumatic brain injury as well as concussion, so looking at mild TBI resulting from sporting injuries and the like. And then we'll look at surgically induced stroke as well. So this is where someone going in for, say, cardiac surgery or aneurysm repair, it's quite common for people to suffer a stroke in those types of surgeries. And I will move on quickly. So we are fortunate to be governed by a fantastic board with so Jeff Pocock, who's here, our chairman, Liddy and Terry, extensive experience in capital markets, as well as an amazing clinical team. So our clinical advisory committee, chaired by David Blacker, there's extensive amount of experience across these gentlemen in running stroke trials. David runs the stroke unit up at Charlie's, so very fortunate. And just some investment highlights, sorry. Are you going to give me a hug?
Do not be sorry, it's a brilliant presentation. Thank you so much for that.
No problem. So just really clear pathway to trials. We are addressing a massive unmet clinical need. We have extensive preclinical data to back up accelerating our drug into the clinic, an exceptional team, and some really fantastic near-term catalysts which is going to drive value in the company. So thank you very much.
Well done.